In MCQ 34, we will investigate genetic engineering and check out the chronological order to introduce a gene that produces fluorescence into an animal itself. So step 1 over here is always to identify the gene of interest that actually gives rise to the particular fluorescence characteristic. And after the gene has been identified in a particular organism, we will remove this gene of interest using restriction enzyme. Now, using the same restriction enzyme, we are going to open a bacterial plasmid. And the act of using this same restriction enzyme actually allowed the gene that was removed as well as the open bacterial plasmid to have what we call as complementary sticky ends. So this facilitates step number 4, whereby when we were to incubate the gene of interest and bacterial plasmid in hopes that it will actually form a recombinant plasmid, so that it's actually inserted the gene of interest into the bacterial plasmid itself. And to help facilitate and catalyze this process, we will be using DNA ligase as an enzyme. So this recombinant plasmid that's ready now will carry this foreign gene that is able to code for fluorescence. And for step number 5 over here, we're just going to use heat and electric shock on a model organism such as E. coli. So what this does is that the pores of the E. coli will actually become transient. It will actually be open for a short and brief period of time. This is where we undergo the process of transformation. Of recombinant plasmid into E. coli. And this forms a transgenic bacteria that's now capable of producing the fluorescence protein via the process of transcribing the fluorescence gene and eventually translating the fluorescence mRNA into that fluorescence protein. So in a chronological order over here, we'll say we definitely have to first identify the gene for fluorescence, excise or to remove it using restriction enzyme. And with that, we will insert that gene into the host DNA, and that DNA can eventually be used for transcribing and eventually translating into the protein for fluorescence. So the answer here would have been 3, 2, followed by 4 and 1, which is C.